Today we are going to see the histology of the male reproductive system. First we are going to see the prostate. Prostate is a very important organ. So much is there in the histology of the prostate, but we are going to concentrate only in the clinical aspects of the prostate and clinical histology of the prostate. Look at this, what type of gland it is. This is the first question usually they ask in the NEET. That is your compound tubulo-alveolar gland. That is the answer. So tubulo-alveolar gland means so many alveoli you can see. You can see, right? So many alveoli. These alveoli are lined by what? These are lined by columnar cells. And you can see basally you have some basal cells. These basal cells, if they get in more in number, okay, if it is increased, that is the only thing how to differentiate it from benign and malignant. So in the pathology slides you can see about the basal cells, and here in the histology of the anatomy you can see the columnar cells. Can you see the columnar cells? You can see the columnar cells and you can see the basal cells here, and in between you have the fibromuscular stroma. So there you have a big nice capsule that surrounds the prostate. The capsule is nothing but it is made up of fibers. So which fiber? Collagen fiber. Usually collagen fiber 1, type 1 only usually we contain in the capsule. Type 1 and type 5 also you can see in the capsule. So capsule is the collagen fiber, made up of collagen fiber, and you can see many number of smooth muscle fibers along the septa. So this is a fibromuscular stroma. In between that you will be having the fibromuscular, the septa is coming from the capsule of the prostate. The capsule, just now I said it is made up of what, and inside that you will be having the fibromuscular stroma. It looks like a road. Through that only the blood vessels are passing through, and here is the alveoli. Each tubulo-alveolar gland is lined by the columnar epithelium, and in some areas you could see some colloid like substance that is called corpora amylacea. That is a glycoprotein like substance. You can see, no, this is called corpora amylacea. We will see about the corpora amylacea later on. Function of the prostatic secretions. What does it contain? It contains a citric acid and the proteolytic enzymes. So what is the role of this proteolytic enzymes? It prevents the coagulation of the sperm. Some sperm should not get coagulated. So what happens? We have a very important factor, prostate specific antigen, which is produced by the epithelial cells of the prostate. What is the role of this? This also prevents the sperm can freely move inside the fluid. Okay, understood? So that is the importance about the prostatic specific antigen and the citric acid and the proteolytic enzymes present inside it. Acid phosphatase, everything. It prevents the coagulation of the sperm in vagina. Then only the sperm can swim. Understood? This is about the prostate. And next thing is prostatic specific antigen. Just now I said the prostatic specific antigen is the important factor which is increased in the cancer prostate. So usually it is produced by the epithelial cells of the prostate. It also the same lytic area, okay, coagulation. It prevents the coagulation of the sperm. And corpora amylacea, just now I said, in old age if you see there is increase in the number of calcified protein collagenous mass. This is called corpora amylacea. What is that? That is present inside the tubulo-alveolar gland. It is a calcified glycoprotein. We could see more in numbers in the old age. You can see the stroma, smooth muscle stroma. And the next is the testis. Testis, you will be having lot of tubules. Okay, seminiferous tubules. In that you can see multiple layers of. Can you see the multiple nucleus? What are these multiple nucleus? These are all different stages of spermatogenesis. The primary type A spermatogonia, type B spermatogonia, and primary spermatocytes, secondary spermatocytes, and these are all called spermatids. Can you see the spermatids? This one is called spermatid. And what is the special cell called Sertoli cell? Sertoli cell action only we will ask. Okay, we need to know about the functions of the Sertoli cell. So one is phagocytosis of the excess. This spermatid is here. Now spermatid to spermatogenesis, sperm, the transition is called spermiogenesis. In those periods, the excess of the cytoplasm material, the remnants of the spermatid, should be digested or phagocytosed by this Sertoli cell, number 1. So phagocytosis of excess cytoplasmic remnants, and it forms a blood testis barrier. This is your blood vessels will be there, and the testis, inside the testis your sperm is there. Sperm acts as an antigen, so the sperm should not be getting any antigen antibody reaction with the blood vessel, blood in the blood vessel. So what happens, it forms a blood testis barrier. And the third one is release of anti-Mullerian hormone. Mullerian hormone is for the development of the female genital system. We don't want that genital system in males. So the factor which is very important is anti-Mullerian hormone, that is produced by your Sertoli cell. Second thing, it secretes something called as inhibin. Inhibin means it inhibits something. It should inhibit what? We don't want the FSH. So it should inhibit the production of FSH, follicular stimulating hormone. So that is inhibin. And production and release of antigen binding protein. Which antigen? Testosterone. Testosterone should be getting, somebody should hold the testosterone, otherwise it will leave out. So who is holding the testosterone? Antigen binding protein, that is produced by your Sertoli cells. And the next one is secretion of fructose rich testicular fluid. So fructose rich means highly energy containing fluid. So it is for the nourishment of the sperm. And the synthesis of testicular transferrin. Okay, synthesis of testicular transferrin. These are all the functions of the Sertoli cell. These entire functions all the students should memorize. Understand and memorize. And then the next one, these are all the tubules. In between the tubules, you will be having these areas, you will be having some cells that is called as interstitial cells. That's why it is called as interstitial. Interstitial cells of Leydig. What is the function of this interstitial cells of Leydig? That secretes testosterone. And then next is we are moving to the epididymis. Epididymis is an interesting organ where the mobility of the sperm takes place, motility of the sperm. Okay, so here also you will be having convoluted, the epididymis is a convoluted tubule-like structure. So if you cut a section, you can see nicely the round or oval shaped tubules. The tubules are lined by pseudostratified columnar epithelium, but not ciliated, it is Stereocilia. What is Stereocilia? We'll see later on. It is a branched microvilli, non-motile or motile. It is not at all motile. It is just, it will be present. As can you see as a filament? It is a microvilli and it is a branched microvilli. You could see the sperm inside that. In testis, the sperm is produced and the sperm is present inside this tubules. Okay, this is sperm. And what is the role of this? Phagocytosis, it has the basal cell and the principal cells. Look at the basal cell. These are all basal cells. These are all the basal cells. What is the role of the basal cell? The basal cells, they have a supporting cell and a stem cell. These columnar cells are called as principal cells. What is the role of the principal cells? The luminal fluid, it absorbs the luminal fluid, and already there in the Sertoli cells, they engulf or phagocytose the remnants of the spermatid. Here the left out remnants will be phagocytosed by this. The most important function, every student should tell this answer. This secretes a substance called as glycerophosphocholine, which acts as a decapacitation factor. Usually where the sperm gets decapacitated, capacitation occurs, the capacitation of the sperm occurs only in the female genital tract, especially in the vagina. But here in the male genital system, it should not get decapacitated. So we need a factor which prevents the capacitation. So that is your glycerophosphocholine, which acts as a decapacitation factor. So keep it in mind, capacitation occurs only in the female genital tract. This is the male genital organ. So it should not get capacitation here. If it occurs a capacitation, it is not much useful. So it has a factor, decapacitation. Then now what is called Stereocilia. Stereocilia is a non-motile branching microvilli. Why it is rigid non-motile? Because it contains actin filaments. That's why it is not motile. And it is a projection from the cytoplasm. And epididymis, you will be having the Stereocilia. What is the action of the Stereocilia in epididymis? It increases the surface area, the epididymis. And what is the use of the Stereocilia in the ear? They function as signal generators. Signal generators means here you will be having the cochlear apparatus. From there you should send the signal. generator. Next slide is the vas difference. Vas difference, already you will be having the vas is a tube. So if you cut the tube, you have the nice stellate lumen. As usual, the epithelium is same, and irregular pseudostratified columnar epithelium with stereocilia, same as epididymis. And the second is the lamina propria, contains collagen and elastic fiber. And the next layer is nicely a big muscular layer, you could say. Why it is so muscular? 1 millimeter thick muscular. That's why easily you can palpate it from the scrotal skin. And why it is thick muscular is because of this contraction only the sperm is moving through that, so it is very muscular. Inner longitudinal and outer longitudinal, the middle circular layer you could see nicely. The last layer is the adventitia, as usual we see in GIT. Next is the seminal vesicle. Seminal vesicle, you can see multiple folds. This is called mucosal folds are thrown. Can you see? So this is called primary fold. From there you have the secondary fold. The folds communicate with each other and they form the lumen. Can you see? They form a circular lumen here. And you have the next layer, as usual, lamina propria, fibroelastic layer you can see, and then muscular layer you can see. Mucosa, primary and secondary folds, and lined by pseudostratified columnar epithelium, short basal cells and tall columnar cells, subepithelial fibroscular layer, and muscular layer is inner circular, outer longitudinal. As usual, you will be having the adventitia. But seminal vesicle produces what? Prostaglandins. It produces prostaglandins. Even prostate also, so many secretions are there. These prostaglandins along with the prostate, it is a component of the semen. Fructose rich seminal fluid, very important. Fructose rich seminal fluid is being secreted. And why the semen is yellow in color? Definitely some chromatic material will be there. What is that? That is called lipochrome pigment. It is secreted by the seminal vesicle. Lipochrome pigment. And it also secretes fructose rich seminal fluid. So this is about the male genital system. The very interesting male genital system. Okay. So thank you.